Hi, everyone. Lots of people from like all over the world. So great to see you all. All right, so it seems like most of the people are in, so we can go ahead and get started. Thank you, everyone, for joining our immunofluorescence workshop today. We're joined today by Sreethu Sankar, who is a product manager here at Proteintech. So Sreethu is a cell biologist by training. He previously worked as an industry scientist for 7 years and has more than a decade of experience in immunofluorescence and microscopy. He is experienced in managing core imaging facility and has several years of experience in confocal microscopy and high content imaging. So as I said, Sreethu is currently a product manager at Proteintech, and he manages all our products for immunofluorescence. So with that, I will go ahead and pass it off to Sreethu so that he can run his workshop. If at any point you do have any questions for him, you can input those into the chat or the Q&A box, and we'll address them at the end. So I'll pass it off to you, Sreethu. Thank you so much for being here.
Thank you, Afrida. Hello, everyone, and welcome to yet another webinar from Proteintech. I hope all of you can hear me well. As Afrida said, I have worked in industry for around 10 years now. I work with some major organization who was invested really in making platforms and reagents around immunofluorescence workflow. So I think I can give you some ideas and insights on immunofluorescence protocols to make your experiment successful. So before jumping into the details of the webinar, I just want to give you a brief introduction about my company. Right now, I'm a product manager at Proteintech. Proteintech is a very well-known company in antibody space. We have around 13,000 antibody targets. We also manufactures ELISAs, IHC kits, and antibody labeling kits. In recent years, Proteintech has expanded into other reagent space by the acquisition of Humankind, which is a cytokines and growth factor manufacturing plant where we manufacture every cytokine and growth factors in human cells, which is HEC293 derived growth factors, which is catering to for your cell culture, stem cell differentiation, and so on. We also have a GMP grade growth factor portfolio, which primarily cater to the cell and gene therapy market. The other company we recently acquired is Chromotech. They are pioneers in nanobody-based technology and reagents. Nanobodies are basically small antibodies, which are like at least 10 times smaller than your conventional antibodies, which is really helpful in some applications like immunoprecipitation, super resolution microscopy, affinity capture, and protein purification. So this is kind of the portfolio of Proteintech. I'll just give you a small brief or story about when these Proteintech started and why Proteintech came into existence. First of all, there's a big problem in the market in the antibody space where antibodies has lack of reproducibility and inconsistency in results. The problem was there was manufacturers who manufacture a product and sell it to multiple resellers. So what essentially happens is resellers buy a product from a manufacturer and they brand it under their name and they sell it. So the problem for the customer is when customer buy an antibody, if an antibody is not working well from reseller 1, and what they do is they go to reseller 2 or the next seller C. So essentially, when they go to multiple sellers, they are again going to get the same products and a different brand name, but there is no way customer is able to figure out that is happening. And eventually, again, the results are going to be messed up. So identifying this problem, our founder and CEO, 20 years back, identified this as a major problem in the market, and we decided that we will manufacture everything in-house so that we have complete control over the quality of the product that we are selling to our customers. So we make sure that we don't buy anything from anybody. We don't sell it to any resellers. So we manufacture in-house and we send it directly to the customer so that we have control over the quality, as well as we can really support our customers in terms of there is any problem that happens to an antibody in terms of troubleshooting. I think customers have really accepted us about our philosophy, and we have seen that this year we are celebrating 200,000 citations for our antibody products this year. And last year, CiteHub is selected as the researcher's choice antibody supplier in 2022. So that's the story of Proteintech. Now, let's jump into the actual topic of the talk today, which is immunofluorescence. Uh, the agenda for today's topic is I will be covering a short introduction about immunofluorescence. Then we will go through the protocols and some tips and tricks for having a successful immunofluorescence experiments. And I'll also talk about some reagents that we are offering to support your workflow. And we can go through a detailed troubleshooting plan and later eventually into a Q&A section. Okay, let's jump into introduction. I know most of the people who are in this call probably are already doing immunofluorescence. This is primarily to those who are just new to this technique. Immunofluorescence as a technique we can explain, and we can actually split this word into two, immuno and fluorescence. Immuno essentially means antigen and antibody reaction, basically. And we are using this antigen antibody reaction to detect or study a protein or a biomolecule in your sample. It may be a cell or a tissue, and you're using fluorescence technology to detect this interaction. So what you essentially do is you take your sample, you use an antibody to detect an antigen in your cell. That antibody is called primary antibody. Now the antigen antibody interaction happened. Second is you use a second antibody or a detection antibody, we call it a secondary antibody, to detect this interaction. Once this has happened, you have to visualize these reaction, right? Now you use fluorescence. So essentially what you do is you use a secondary antibody that is tagged with a fluorophore. You use that fluorescent tagged secondary antibody and you use an excitation light, a light source from your microscope to excite and visualize this reaction. So what this reaction tells you is basically the localization or the expression of specific protein or a biomolecule in your sample. So that's about immunofluorescence as a principle. Now, coming to general immunofluorescence staining protocol, there are some variations you can find for these protocols according to different samples, but this is primarily a general protocol that people use it for cells and tissues. All right. So there are basically 8 steps involved in, once you receive your sample, the first thing you do is fix your sample. Essentially, you want to arrest your cell in a specific metabolic state to study it, right? So for that, you do a process called fixation. Fixation involved use of different chemicals. Most commonly used one is paraformaldehyde. And then organic solvents can be used for fixation. Once fixation is done, you wash your samples, and then the next step is to permeabilize your sample. Permeabilization isn't so you have to use an antibody in the later step. Antibodies are bulky molecules, so you need to really pay or clear the way for antibody to get inside your cell. For that, you need to do permeabilization. Permeabilization is essentially done using a detergent mostly, usually Titon X100, and there are some other reagents like saponin which can be used for permeabilization or making way for your antibody to enter the cell. After permeabilization, the next step is blocking. Blocking is done to avoid any non-specific binding of your antibodies, can be primary and secondary antibodies. Blocking is usually done using serum or bovine serum albumin. After blocking, it comes the addition of primary antibodies. Primary antibodies are added to your sample and you leave it for some time. That also protocol varies. It can be 4 degrees Celsius overnight or 3 hours at room temperature. For this, you're giving enough time for this antibody to interact with your antigen and bind to it. After that, now you have to do a thorough washing of your sample and then followed by addition of secondary antibodies with some fluorophores onto it. So after secondary antibody is added and you give it time for incubation, you can choose to add some subcellular marker stains or some counter stains, which might differentiate between different structures in your cell. It can be either a nuclear stain or a cytoskeletal stain or something like that. Post that, you can use an anti-fade mounting media or any mounting media to mount your sample for before you take it for microscopy. So this is the general IF staining protocol. Now let's look at step-by-step considerations in this protocol. So first of all, I think I started with the step of fixation. I think there is one most important step even before fixation is consideration about your sample itself. I think importance is you need to have a basic understanding of your cells or tissue even before starting your experiment. It's very critical for a successful immunofluorescence. If you have a basic understanding of your protein function and potential distribution in the cell, you can optimize your different protocol step according to your sample. Second thing is protein expression and distribution is influenced by cell health and confluency. So when I say cell health, as you know, when people are doing cell culture is prone to a lot of different contaminations. Primarily, maybe bacterial contamination, yeast contaminations, and so on. But there is a silent killer which is being overlooked all the time. I think it is mycoplasma. Mycoplasma is a contaminant which we can't really see with our regular microscope or just by looking at 120x or 40x objective, because they doesn't have cell wall, so you don't get to have a contrast to see it, and they're so tiny. But the impact of mycoplasma on cells is can be really bad and can definitely affect your results. So I can show you an example. So on this image, which I am showing here, this is HeLa cell on the left-hand side, which is a mycoplasma negative HeLa cell. On the right-hand side is a mycoplasma positive HeLa cell. The key difference here, I have used phalloidin, which is a cytoskeletal stain, to stain both the cells. Now, if you look at mycoplasma negative cells, you can clearly see that the cytoskeletal structure are well organized and continuous. But however, in mycoplasma positive cells, you can see the cytoskeletal structures are completely broken. So as you know, the cytoskeleton is really essential for maintaining the cell morphology, and also there are many other proteins which are associated with these structures. So it is so important that you make sure that your cells are free of mycoplasma. There is no clear study of what all way mycoplasma can impact your results, but definitely it can have these broken cytoskeleton, it can damage your cell morphology, it can also cause protein, antigenic reorganizations and so on. So I think the best way to do the detection of mycoplasma is using a PCR. It is very sensitive technique which can detect mycoplasma in a really early stage, ideally. And if your cells are highly infected with mycoplasma, you can also visualize this using some DNA binding dyes. Now, next thing I told about is cell health, and second one is confluency. How cell confluency or how cell density influences your protein expression. So there are several proteins which is the expression of proteins are influenced by how confluent, how dense your cell has grown. Here is an example. Okay, so this is a protein called occludin, which is a tight junction protein. I have tried to visualize this protein in cells with two different confluences. This is same MCF7 breast cancer cell, and using same antibody. On the left-hand side, you see a cell which is 50 to 60% confluent, maybe even less. And the right-hand side, it is 80 to 90% confluent cell. When this protein especially is a tight junction protein, you need to really give cells to form this time for the cells to form junctions, and which will only occur once a cell reach an optimum confluency. So 50 to 60% confluency may not be right amount for the specific protein. So it is important to make sure or understand the level of protein expression in your cell, or basic understanding of how much the protein expresses depends on your confluency of cell. The third important point is dealing with tissues. So make sure to select tissues that are not folded or damaged. What I mean by this is usually when you take use a tissue to section in a cryotome or or a cryostat or a microtome, essentially what happens when you go for really thin sections, there is chances that your sections get folded, and tissues are inherently uh, autofluorescent nature. So once you have folded sections or damaged sections, there is a good chance it will eventually affect your final results. And there might be a lot of autofluorescence from a specific region you are interested in. So it's better to make sure, even before starting your experiment, observe your tissues and make sure that the tissues are not damaged or there is no folded part in your tissues. This is about the importance of looking at your cells or tissue even before starting with the fixation. Now, let's look at fixation. As I said, fixation is a process to metabolically arrest your cell in a specific stage, right? So there are different types of fixatives. One is the most common one is paraformaldehyde. It cross-links proteins and biomolecules through formation of methylene bridges. It conserves cellular morphology best, even for tissue, it is really good, but it can actually mask epitopes and cause autofluorescence. So it is very important to kind of optimize these fixation time and protocol for your sample. And when it comes to paraformaldehyde, it's always recommended to freshly prepare paraformaldehyde because they are really unstable in nature. Although there are protocols which mentions that you can prepare paraformaldehyde and freeze it for some time, but I would always recommend to use it fresh. If not, if you're freezing it, don't keep it for more than 3 weeks or a month. So some protocols recommend you can keep it for more time, but I wouldn't really recommend it to keep it for really long time as it is really unstable in nature. Uh, Next, as most common method of fixation is organic solvents, which is ethanol or methanol or acetone. It doesn't mask epitopes. It works by precipitating your protein. It also delipidates your membrane. It dissolves the lipids. Here is an example on the right side where two proteins with different fixatives, how it performs. And number one is STORM20, which is a mitochondrial protein. We've tried to fix it with both PFA and methanol. You can see that with PFA, it works absolutely fine. And with methanol, the protein is not. E-cadherin, on the other hand, which is a membrane protein, when you fix it with paraformaldehyde, it actually causes an antigenic redistribution. And this, it seems to be we are detecting something on the nuclear membrane and in cytoplasm. Whereas when you fix it with methanol, you actually get a proper membrane staining. So the crux of the story is a different fixative can influence epitopes differently. Nobody really knows. There is no standard here. So to follow like what fixative to use for what protein. Essentially, at least whenever you're starting with an experiment, a new antibody, always try at least two different fixation methods, one with paraformaldehyde and one with an organic solvent to make sure that which fixation method works best for your antibody and your protein. And coming to some other samples like organoids and spheroids, which are thicker samples. So these samples need more time for fixation, for the fixative to go inside the organoid structure, and it has a lot more extracellular matrix and all. So you need to give more time for organoids and spheroids to fix. Usually the recommendation is around 45 minutes or sometimes 30 to 45 minutes for fixation. So, but again, for your sample type, you need to kind of optimize these conditions. And that's about fixation. Now, as I said, one problem with paraformaldehyde fixation is masking of epitopes. So this essentially happens more in tissues, because tissues are more complex in nature. In order to revive these epitopes back, there are methods called antigen retrieval or epitope retrieval. There are two methods which are commonly used. One is heat-induced, and second one is proteolytic-induced epitope retrieval. Heat-induced epitope retrieval is the most commonly used method. It's actually really gentle. which uses a low pH buffer and heat your samples to retrieve your epitope. And there is also another method called proteolytic, where you use some proteases to digest some of these ECMs and crosslinks so that you can access epitopes. So these are the two methods of epitope retrievals, especially when you're dealing with tissues. Next step in an immunofluorescence protocol is permeabilization. It is done for accessing intracellular targets. You're basically making way for your antibody to enter the cell and bind to your protein of interest. The conventional way, the most commonly used method is using detergents like Triton X100. The recommendation is from 0.5 to 0.5% for 10 to 15 minutes. These are really strong detergents, so we always need to make sure that you don't over permeabilize it or keep it for a longer time. You will end up losing a lot of epitopes. In case if your targets is just cytoplasmic or sometimes membrane, you don't have to go for harsh detergents. Instead, you can use milder detergents like saponins or digitonin, which is milder saponins, which essentially removes a cholesterol molecule from your membrane and which makes way for your antibodies to enter your cytoplasm. If organic solvents like ethanol or methanol or acetone is used, no further permeabilization is required because organic solvent itself can delipidate your membrane and make holes in the membrane. So you don't have to further do a permeabilization step for samples fixed with organic solvents. For FFPE tissues or formalin fixed paraffin embedded tissues, permeabilization is again not required because these tissues go through multiple stages of treatment with organic solvents, which is not required. For some samples like thick tissues and organoids, the length of permeabilization needs to be optimized. Especially for organoids and spheroid, you may need longer permeabilization time. Usually it is around 45 minutes or 30 to 45 minutes, but that also need to be optimized based on the thickness of your sample. Next is blocking. Blocking is done. So antibodies can interact with other biomolecules in a specimen or in a sample. So blocking is done to prevent these non-specific interactions, because if you have non-specific interaction, by the end of your results, you will start seeing a lot of background in your sample. In order to avoid this, use some blocking buffers. Usually the most commonly used blocking buffers, or the recommended one, is serum, which is from the host species of secondary antibody. The concentration of, or the percentage of serum that is being used for blocking is usually ranges from 2% to 10% for around 45 minutes. Bovine serum albumin works very well for your blocking. It's also widely used. 2 to 5% of BSA works very well for blocking in immunofluorescence. As I said, blocking reagents again interact with these non-specific sites through weak interactions. You don't really have to wash the sample after blocking. Even in previous steps, like after fixation, after permeabilization, you have to wash your samples, but after blocking, you don't have to wash your samples, because washing samples may just wash out this block site as well. So that can be avoided. Next is the most important step in your entire experiment, I would say, primary antibody selection. First of all, the most important information that everyone needs to understand is not all antibodies are suitable for all applications. So when a manufacturer makes an antibody, it doesn't work the way that we really want, because we always want an antibody to work in all the application, but it's not that way. That's how it works. Some antibodies recognize linear epitopes. Some antibodies tend to recognize conformational epitopes. So because of this reasons, it's not very easy to find an antibody that works in all applications. So on the right-hand side is an example. This is our own antibody, ASGR1. On the top, you can see an antibody against ASGR1 protein that works across multiple applications like Western blot, IHC, and immunofluorescence. On the other hand, there is a second antibody in our portfolio, which we have developed specifically for Western blot, which doesn't work in immunofluorescence. So it's extremely important to look at the supplier's website and find that if this antibody is actually developed for your intended application, here specifically immunofluorescence. That's really critical. So, and you need to always optimize your antibody concentrations and incubation time. Usually recommendations are like 3 hours in room temperature or overnight in 4 degree are the recommendations for antibody incubation times, which again depends in my hands. 4 hours was always better because I think in lower temperature, the reaction is kind of slow. So it will allow only specific interactions. And in lower temperatures, you get much better binding efficiency and a very less background. About antibody concentrations, a supplier always recommend a concentration. That concentration recommendation is based on the samples they have tested. The samples that you are using might be very different. So always take some time to optimize antibody concentration specifically for your sample. So I think a good starting point will be from looking at the supplier's website and maybe do a dilution, maybe 2 dilution above and 2 dilution below what they are recommending to find out what is best for your sample. Next is secondary antibody, which is after primary antibody, you add a secondary antibody for detection. So selection of secondary antibody is also as important as primary antibody. The first thing to note for secondary antibody selection is host species and isotype compatibility. You need to always make sure that your secondary antibody is raised in a different species than which the primary antibody is raised, then only it will react. Consider the isotype of the primary antibody. So there's a lot of mouse monoclonal antibodies out there in the market, and it comes in different isotypes, like usually IgG1, IgG2A, IgG2B, and so on. Now, if you are selecting an anti-mouse secondary antibody, which is specifically developed against IgG2A, but you are essentially looking at our primary antibody has an isotype of IgG2B, both comes from mouse, but your secondary antibody that you have selected may not work for your primary antibody. So it's always important to understand your isotype of your primary antibody prior to selecting your secondary antibody. Fluorophore match with your microscope filter. When you're performing immunofluorescence, different labs, different people use different settings and different filter settings for your microscope. So always see if the fluorophore that you're selecting is compatible with the microscope filters that you have. That's a very important things to consider. And next is cross-reactivity to tissue or species. This is specifically or especially important when you are doing multiple labeling experiments. If you are adding 2 different antibodies or primary antibodies from 2 different species, say mouse and rabbit, since these animals are physiologically very similar, the secondary antibodies have a chance to cross-react between each other. So if you add a mouse and rabbit together and you buy an anti-rabbit and anti-mouse antibody, which has a tendency to cross-react, you may not get the results that you want. So what you have to do here is you can always look for cross-adsorbed and secondaries, which means, excuse me, these secondary antibodies are cross-adsorbed against some other species like rabbit or rat, so that it doesn't cross-react with that kind of species. Again, like I said, for primary antibodies, always optimize the dilution and incubation time for secondary antibody as well. Fluorophore selection, there are traditional dyes available like FITC, Texas Red, and all that. So these traditional dyes tend to bleach easily. Although they are fluorescent, they are sensitive to pH and not that bright. The new generation dyes like Alexa Fluors and Coralites, which are in fact sulfonated dyes, which is extremely powerful in terms of their brightness and pH sensitivity, and it lasts really longer. doesn't bleach that faster. So always make sure, consider to select some new age synthetic dyes for your immunofluorescence. The last step in your protocol is mounting. Why we have to do mounting? First of all, it can reduce photo bleaching. You can use your sample for long-term storage. Your image quality can be increased. So uh, how it improves the image quality? Usually after all these processing of your sample will have an uneven refractive index basically. So your mounting media can evenly give a refractive index to your sample, and this refractive index will be matched to your objective of your microscope, so that your microscope objective is able to capture a maximum signal from your sample. So essentially it will help to get a much clearer and much better result image. Most commonly used is aqueous or glycerol based mounting media. Excuse me. There are hard setting mounting media, which is better for long-term storage. Hard set mounting media, you need to be really careful when handling hard set mounting media, because hard set mount, once you add it to the sample, you need to give enough time for this mounting media to set, which is called curing. Usually you can keep it in overnight, so it cure and become set and hard and more like a solid. Once it reaches that stage, then only this mounting media will show it actual properties. That means uh, the mounting media will reach the right refractive index once it is set. So we need to always make sure that your hard set mounting media is properly set. Next is anti-fade. Many of the mounting media comes with anti-fade reagents, which are free radical scavengers, contains free radical scavengers. So photo bleaching happens because of reactive oxygen species. So anti-fade reagents can help scavenge these free radicals. But keep in mind that some of these anti-fade contents can have an effect on your fluorophore itself. And also it's important to note all mounting media is not compatible with all fluorophores, because again fluorophores are chemicals, mounting media is also chemical. As I said, fluorophores are sensitive to pH and other conditions. Once you have mounting media that has different components, yeah, sometimes it has or it reacts with your dyes and you can quench the dyes. So there are a lot of tables and charts available online. You can look at what mounting media goes well with your fluorophore of interest. um, Now I'm moving to um, immunofluorescence, direct immunofluorescence, and the difference between direct and indirect immunofluorescence. In right now, we were all this while I've been talking about indirect immunofluorescence, where you use a primary antibody followed by a secondary antibody. There's also an option to do direct immunofluorescence, where you can completely exclude the use of secondary antibodies. You can buy an antibody which is already a primary antibody, which is already conjugated to a fluorophore. So it's much faster, it's easier. I'll tell you what's the reason to go direct. It is lower background, no off-target binding from secondary antibody. You don't have to think about secondary antibody because you're not using it. Perfect for same species. That means, especially when you're handling mouse tissues, there is a chance that your mouse tissue has endogenous IgGs, and then you add a primary antibody, a mouse primary antibody, followed by an anti-mouse secondary. These secondaries can react with the endogenous antibody as well. That can create a lot of noise or background in your sample. So you can avoid all this if you go direct. It is much faster, simpler. It is easy for multiplexing. Again, if you want to study or do a multiplexed immunofluorescence, always better to go with for direct method, because you can use multiple antibodies without even considering what species it comes from. It can be all 4 or 5 comes from mouse itself. It doesn't really matter because it's already conjugated. So these are some of the advantages of going direct. So I think we have covered the important part of protocol. Now I will introduce you to some key reagents that you can look for your immunofluorescence workflow. As a company, we try to offer a complete workflow for immunofluorescence, which starting from growth factors for your cell culture. As I said, we have humankind growth factors, which you can use for your cell culture or stem cell culture or differentiations and so on. Then we have primary antibodies, which has both polyclonal, monoclonal, and recombinants. Then from that, we have directly conjugated antibodies, secondary antibodies, nanosecondaries, nano boosters for signal amplification, trauma bodies for life cell imaging, and we also have a new addition to our family, which is flexible antibody labeling kit. I will talk more about this in the coming slides. Primary antibodies, we have both polyclonal, monoclonal, and recombinant. All of these have its own advantages and disadvantages. Polyclonals are ideal for low abundant targets and mutated proteins and so on. Polyclonals, monoclonals, it's better for high abundant targets because it has much low background. Recombinants are much better in term for long-term projects because it is highly reproducible in nature as it is coming through recombinant method. Disadvantages for polyclonal, there is a chance of high lot-to-lot variability because it's directly isolated from animals itself, and you can't always expect animal to respond the same way. That's one disadvantage of polyclonal. Monoclonal, conventionally, epitope accessibility is low compared to polyclonals, and there are issues with mouse on mouse, as I said in my previous slide. And recombinants, target range is again poor. So this is a directly conjugated antibody portfolio, which is called Correlite primary conjugated antibodies, specifically or exclusively developed for immunofluorescence. We have a portfolio of 2,000, more than 2,000 conjugated antibodies, which is available in 4 different colors: 488, 555, 594, and 647. Correlites are one of the brightest and most photostable dyes in the market. So we have used these dyes to develop so what we do is we select our own antibodies which are already cited for immunofluorescence, and we conjugate it to these dyes to make it easy for immunofluorescence. So what I'm showing here is some examples of data generated by our own customer. From studying neuromuscular junction to mouse hindbrain and mouse spinal cord using beta-3 tubulin, which stains your neurons, and GFAP, which is a marker for your astrocytes. We also try to generate multiplex image panels to support cancer immunology research. This is an image of some key markers expressed in colorectal cancers. So these all are again directly conjugated antibodies. We try to stain the cancer cells as well as immune cells, primarily T cells and cytotoxic T cells, using conjugated antibodies. Next important reagent I really want to introduce here is flexible antibody labeling kit. This is actually one of its kind product in the market, because antibody labeling conventionally is a really time-consuming process, and it is really tricky to master that process. Now we are coming up with this product to make it simpler, and it is easy to use for anybody. First of all, this product is compatible with any primary antibody from any vendors in the market. And you can label your primary antibody in 10 minutes, just 10 minutes. And if you look at conventional labeling technique, it takes hours for labeling. It has to go through purification step, it has to go through buffer exchange step, and so on. Here, you don't have to do any of this. You can start with as low as 0.5 microgram of antibody, but for a conventional technique, it's at least you need 500 micrograms, 100 times more antibody. Here I'm talking about 0.5 microgram of antibody you can start labeling. You can label many different antibodies with one kit. So essentially what we do is, like, you take an antibody. This is a proprietary polypeptide that we developed, which can bind to antibodies. This polypeptide has a fluorophore conjugated to it. So once you add this polypeptide to your antibody of interest, it'll just go and bind to your antibody. Now you can use this antibody for all your experiments like immunofluorescence, flow cytometry, and Western blot as well. So how it works is basically you have to take your antibody, you have to add the flex linker in the previous slide. The polypeptide is called flex linker. If there is any unbound flex linkers, we can always quench it using flex quencher. That is another technique that we have developed. So the step-by-step, if you want to look at the protocol, what you basically do is you take your antibody of interest, you add your flex linker, incubate for 5 minutes. Now you add the flex quencher, incubate for 5 minutes, 10 minutes, you're done. You're ready to image or you're ready to do your experiments. So this is how it works. And especially this is really helpful even for multiplexing experiments, especially you can't always find, good primary conjugated antibodies for all the targets that you're interested in. Here, this flexible kit will solve this issue because you can take, your antibody of interest. We have flexible available in multiple colors. Use the same way, add it, incubate for 5 minutes or 10 minutes, and then you mix all your antibodies, which is labeled with different colored flexible. Then you add it to your sample. It's ready to go for multiplexed imaging. So it's so easy. So it's actually, a really fast method to do multiplexing or just labeling your antibodies. Next reagent I want to introduce you is about nanobodies. Nanobodies are, again, as I said, it is as 15 kilodalton small antibodies, which is actually or isolated from camelid species. They express these small nanobodies. This has a lot of advantage because of its smaller size, and it can penetrate tissues much better because of the smaller size. And this is an example on the right hand side, which one nanobody based reagent called nano boosters, which is used for boosting the fluorescence of some of the proteins. So here it is GFP booster. So you can, sometimes the GFP expression might be too low or it might be faint. You can use this booster to boost the signal of GFP in cells or tissues. Even in whole animal imaging, you can see how it is done. It is also, the size is too less. So there is a problem in super resolution microscopy called epitope label displacement, where when you have two normal antibodies, because of the size of the antibody itself, there is a displacement of your label, which can essentially affect the resolution of your final image. So if you use nanobodies or nanosecondaries or nanoboosters, you can kind of get rid of this. It's much better for increasing the resolution, super resolution microscopy. These are some examples of how nanobody based reagents are being used in super resolution STED microscopy. This is a spot label on the left side. On the right hand side, it's a Wyminton label, which is used for super resolution. This is just an example to show you how you can actually utilize the entire workflow from Proteintech. And this is just a differentiation of pluripotent stem cell to definitive endoderm using our own growth factors like activin A, Win3A, BMP4, and so on. After differentiation, we use our primary antibodies like FOXA2 and NANOC to stain it. Then counter stain using flexible kit or primary conjugated antibodies and our correlite conjugated phalloidin. Now, let's jump into the another major part of my presentation, which is troubleshooting. So first thing I have to talk here about troubleshooting is about controls, because I think if you have right controls in your experiment, including controls will let you decide on where exactly things are going wrong in your experiment. So for shooting the trouble, you need to first find out what the trouble is. So controls are something that really helps to do that. So these are the 4 different controls which we have to think about when you start immunofluorescence experiment. First one is secondary only control, which basically ensures that your signal is coming from primary antibody. If you add a secondary or antibody alone to your sample, and if you still see some signal, which means the data that you're getting, you need to take it with a pinch of salt, because your secondary antibody might be non-specifically binding somewhere. So that is one control. Next is endogenous control, which is without use of any antibody in your sample, no primary or no secondary antibodies. This kind of reveals the level of autofluorescence or artifacts in your sample itself. And if possible, you have to add positive control and negative control to validate your results. Positive control can help you find out if you're actually, whatever sample you're seeing is right. The negative control reveals false positives. So at least in your experiments, always try to include a secondary only control and endogenous control to make sure that whatever you're seeing is right. So other major problem with, especially when handling tissues, is autofluorescence. Autofluorescence can be caused by two reasons. One is fixation-induced autofluorescence. Second one is autofluorescence from endogenous molecules itself. So fixation-induced autofluorescence happens especially when you're fixing with paraformaldehyde, which can leave a lot of free aldehyde molecules, which is really autofluorescence in nature. In that case, one tip to fix it is to add a small concentration of glycine, which is an amino acid. It's like recommended is 0.1 to 1 mole of glycine in a blocking reagent. So this glycine can actually go and bind to this aldehyde groups, which will reduce autofluorescence. And most autofluorescence happens in this 300 to 400 nanometer range. So use far red dyes like, correlite 594 or 650 so that you can reduce autofluorescence. Red blood cells are again a major cause of autofluorescence. You can perfuse with PBS to remove RBCs before even fixing your sample. So the perfusion with PBS can help in getting rid of RBCs. Reagents like Sudan black and sodium borohydrase is also help in reducing autofluorescence for certain channels. Although a slightly unconventional method, you can pre-expose your tissues or with high intensity UV lights to reduce autofluorescence. So it's basically you are trying to bleach out the existing autofluorescence using high intensity UV lights. Although this is not really commonly used, but it is really efficient. The problem is it is difficult to find these high intensity UV lights in most labs. That's probably the reason why it has not been widely used, but still people use it and find it really helpful in reducing autofluorescence in tissues. Next is weak or no signal, which is something that often everyone who does immunofluorescence encounter. The potential cause would be one is your primary and secondary antibody. You have to have a check on it because of the storage or long-term storage, this might have gone bad. So you can always check whether these things are working or not. If it is not working, you can sometimes it just the antibody just get degrades. So you can get rid of that antibody and have to start a new one. Antibody conditions not fully optimized. As I said in my previous slides, sometimes you need to always optimize the concentration of your antibodies because your sample may not be expressing the highest amount of protein. So you probably have to add more antibodies. So that condition needs to be optimized according to your sample to get proper results. So protein of interest not expressed, that is another common problem where your sample is not expressing the protein of interest that you're looking at. So running a positive control along with your in your sample can kind of find out if that's a reason. If that's the case, probably you need to think about what sample you need to be using. And if your protein of interest is low expression, you can use signal amplification methods. There are different methods, like we recommend nano boosters for amplifying signal. Sometimes epitope accessibility is a problem. After fixing with paraformaldehyde, the epitopes get masked. So you can try using different epitope retrieval methods to regain some of these masked epitopes. So it will eventually work. Troubleshooting, again, next is high background, which potential causes is primary or secondary antibody concentration is too high. Titrate your antibody concentration, decrease incubation times. So right side, you can see an example of FBL antibody. Uh, you sometimes we just try to think that you add more antibody, you get brighter signal. That's not always the case. FBL is a nucleolar protein. If you see 1:100 with 1:100 dilution, you get signal in both nucleolus and in around the cytoplasm and everywhere, which is essentially nothing but background. Once we reduce the dilution to 1:500, you started seeing much crisp staining of nucleolar structures. So this is just an example to show how these dilutions of antibodies so important to get proper staining in your immunofluorescence. Non-specific antibody binding can happen maybe because of blocking, or sometimes it's just because of antibody itself. If the antibody is not well characterized, it has non-specific interaction with other similar proteins or molecules in your sample, which can cause a lot of background. Endogenous IgG dealing with tissues. If it is endogenous IgG, your secondary antibodies interact with this endogenous IgG and cause a lot of background. Which can be avoided by using some mouse-on-mouse reagents, which can block this endogenous binding, or using primary conjugated antibodies. Sometimes poor washing of samples can cause high background. So prolonged washing step, or maybe washing it multiple times. I would always recommend to wash like 3 to 5 times, and every washing step, like 5 minutes on a slow rocking is really helpful. And slides drying out can also cause high background. Never let your slide to dry out. When you're incubating your slides or samples, always use a humid box so that your samples are not evaporating faster. So these are some helpful tips to troubleshoot your experiments. So in conclusion, what I want to tell you is for a successful immunofluorescence experiment, first of all, be patient, choose the right models and antibodies. Always optimize your fixation, permeabilization, antibody concentration, and incubation time. Use all necessary controls. So if you follow all this, I think you can always win with immunofluorescence. That's it from me. Thank you so much. Thank you for listening. I think we can move to the Q&A now. We have a lot of other resources in our website, which you can refer to for different techniques. Also, before jumping to the Q&A, I'm again the product manager for immunofluorescence. You can reach out to me as well. We are also running a microscopy immunofluorescence competition right now. If anybody of you are interested, please enroll. You don't have to use Proteintech product or anything for the image. You can use product from any supplier. You can enter. There's absolutely grand prize for this competition. And we have a gift card. You will get a printed image of your own image itself. So it's a great platform. And thank you for listening. I think we can move to the Q&A.
All right. Sounds good. Thank you so much, Sreethu. That was a really informative, great talk. It was really amazing seeing all the wonderful questions coming in, and not only that, but people were answering questions and it became a discussion. It's really amazing to see, scientists helping other scientists and sharing protocols. So, thank you guys so much for really having this community and sharing your tips with each other. I do want to just do some of the questions that we have. So Sreethu, there were a few questions asking about, how our correlites compare to Alexa Fluor. Can you expand a little bit on that?
Yeah, absolutely. So correlites we have done some studies. So correlites are very similar in structure. Excuse me. Similar in structure when compared to Alexa Fluors in terms of brightness, and its photostability is also very similar or sometimes better. I would say, because we have used some cool technology in our correlite dye making. We try to make the dye as hydrophilic as possible using a technology called adding pegylated molecules, which makes it really hydrophilic in nature. So It compared to Alexa-Flor, I would say it is equal or better.
Great. So someone also asked, can you skip the permeabilization step if you want to capture surface molecules of interest?
Yes, you can. It depends. So sometimes your antibodies may be recognizing an epitope of your surface of your maybe a transmembrane protein. If you take your epitope might be inside your cell. So if you have understanding of where exactly your protein resides, you can always skip the permeabilization step, or you can even use a very mild permeabilization in case if you're doubtful about an intracellular domain having an epitope.
Great. So there's also another question about specifically how does blocking work? So how does it support specific antibody recognition?
Well, it's a very interesting question because blocking works by any blocker. If you add blocker, just bind to bind everywhere non-specifically. So now once you add the primary antibody, which is a specific antibody, the primary antibody will compete with the blocker. And since the primary antibody is more specific to your epitope, it's a competitive binding. So it just displaces your blocking agent and just bind there. So that's how a blocking works.
Great. Thank you for
Yeah.
that explanation. Moving on to the next question. So there was a question about cell confluency. So if they have a cell confluence of 80 to 90%, could this cause false positives in their staining?
Not necessarily, it depends. Again, it depends on what protein you are looking at. So my point is always ensure you don't have to go for an 80 to 90% confluency for every protein that you're studying. So you need to have an understanding of what protein you're looking at, especially for protein that is in junctions. You probably need that kind of a confluency. Okay, so it again depends on what proteins you're dealing with.
Okay, great. So kind of moving on to, how to kind of find the right controls. There's a specific question that was asking about, if you use paraformaldehyde, can you solve the autofluorescence problem by making a negative control with only your secondary antibody and then essentially subtracting that negative control from your experimental condition?
Negative. Okay, can you repeat that question again?
So essentially they're asking, can you avoid the autofluorescence problem by using a condition where you have just the secondary antibody only with no primary antibody?
So for autofluorescence, you don't have to use any antibodies. You can just exclude both the antibodies. Autofluorescence is the property of your tissue itself,
Okay.
or is it maybe because of fixation induced? So you can just use your tissues and you can subtract it. But the problem is sometimes autofluorescence is too high that it might even mask the actual signal itself. So in some tissues, there's really high amount of connective tissue, or some tissues, if it is not processed well, you have a lot of RBCs in there. This can cause autofluorescence, which can hinder with your actual reset itself. But yes, up to a certain level, you can negate the amount of autofluorescence using some algorithms.
Okay. Yeah, and I saw in the chat there was or the Q&A, there was another question that was asking, how do you know that you have autofluorescence?
Mhm.
So would the answer to that just be not use any antibodies and see what your tissue looks like?
Exactly.
Okay. Um, so there was also a question, someone asked, can you use conjugated antibodies of the same host in one sample?
Oh yes, of course. That's again an advantage of your conjugated antibody. You can use 2 or 3 or 4 conjugated antibody at the same time from the same host. You don't have to worry about host at all.
Okay, and then what about in a situation where you have one antibody that's conjugated followed by another primary mouse that requires a secondary? Is that something that you could also do?
Yes, of course, you can do that. You can combine conjugated and unconjugated antibodies together. But when you do that, again, you need to just think about what species you are using, because there is a chance that your secondary antibody will bind to your conjugated antibody as well.
So is there like do you recommend doing one first before the other? Like, should you do the primary and secondary first before you add the conjugated, or?
There are different protocols. You can do that if you are using a different species. Like say you are using antibodies from conjugated antibody from mouse, and your unconjugated is from rabbit, then you don't have to worry about it. You can mix it together and use. But if not, you can just do simultaneous staining of conjugated and unconjugated.
Great. And then there was also a couple of questions kind of related to that in terms of, can you do something similar using flexible? So conjugating with flexible and then doing a primary and secondary antibody staining after.
Yes, you can. Yes. So the advantage of flexible is you don't have to really worry about it. We have flexible available in multiple colors, and you can use it with all different antibodies. So you really don't have to worry about adding a secondary antibody. But if you want to do that, you can always use that as well with flexible, because you don't really have any unbound flexible available to interact with any other antibody, because it's already quenched with flex quenches.
Perfect. So we have time for just a few more questions. I know we're going a little over, but there were so many questions. I want to make sure that we hit a good chunk of them. Is there a particular blocking agent that you would recommend if you're trying to detect phosphoproteins?
Usually, BSA works very well, um, but some recommendations are not to use PBS, because it sometimes interact with your phosphor form. But I'm not very sure. I have read about it, but I haven't really experienced any problem with that. But for phosphoproteins, BSA itself works very well. People have tried casein separately for that, but it was not really successful. Some people recommend to use casein, but it's not been very successful in my hand.
Yeah, I was going to say, I don't think I've ever looked at phosphoproteins through IF, but yeah, I feel
Yeah,
like,
but people do look at it, because I think sometimes is after phosphorylation, some proteins tend to move around. It changes its localizations. So for some proteins, people do study it using immunofluorescence. But yeah, general BSA or your serum usually works well.
sounds good. Um, so would it be detrimental for staining if you blocked for too long? If you block and then go to lunch and you're like, oh shoot,
No,
it's been a
it's
few
not really.
hours.
Not really. Well, in fact, you can even store your samples in blocking buffer. It doesn't really have any big impact on your samples, as far as I know. Just need to make sure, if you're really storing it for a longer time, you need to make sure that you add, because usually blocking contains a lot of proteins and amino acids, and also it can be a good place for bacteria to grow. So if you have to always make sure that uh, you don't leave it there for a longer time, and you're keeping a sample for a longer time in blocking. If you're doing that, you just need to add some acid or something, but it doesn't really affect your experiment at all if you keep it for long.
So during your presentation, you made a big emphasis on, using fresh PFA.
Mhm.
Um, However, there are certain companies that also, sell PFA. So what are your recommendations or thoughts on using PFA that's been purchased from a company?
You can use that. I think in order to stabilize PFA, many companies add a small amount of methanol in PFA. So you need to think about, so there are methanol infused PFA that is the most commonly available to stabilize PFA that is available. If not, PFA comes in ampules, which PFA is again light sensitive. It comes in dark ampules, where they say it's usually one time use, just use it and throw it. So I think, and it is not interacting with anything, air molecule or anything, for it to decompose. That's why it comes in ampules. So for that kind of storage or that kind of packing, it is okay to store it for slightly longer time. But if you're preparing it by yourself, always recommend to prepare fresh.
Okay, sounds good. So I think we have time for maybe two more questions. So one question is about, they were specifically talking about organoids, but I think it's a good topic to just cover in general. What would happen if you fixed your tissue or your organoids for too long? What could you expect to see?
So too long, like depends on how do we define too long? Because organoids and tissues require longer fixation times. And like tissues organoid, if you fix it for too long, one major problem is if you're using paraformaldehyde, it fixes through cross-linking. Over cross-linking can mask a lot of epitopes. That is one major issue that we will face, especially when you have a lot of extracellular matrix proteins. It can definitely mask a lot more, epitopes. So you need to always figure out, especially if you're studying multiple proteins at the same time, you have to figure out ways to retrieve these epitopes back. And it always won't work for every epitope that you want. So it's better to optimize that time instead of fixing it for too long, I would say.
Sounds good. So one last question. So it's more about, your preferences of your protocol for IF. So when you do washes, do you use PBST throughout the entire protocol, so after permeabilization? And if so, do you have the same amount of Triton X100 concentration as the solution from permeabilization?
Well, so PBST after permeabilization, I usually use Tween 20, not Triton. And Tween is a milder detergent. So that is being used. Post permeabilization, every step, I always try to include a small amount of Tween 20 because I feel that if there is any small non-specific interaction, these detergent presence can kind of wash away these non-specific binders.
Okay. And then in terms of, kind of similar question, when you're doing blocking, do you keep the same serum concentration between blocking, primary incubation, and secondary incubation?
So for blocking, usually the serum concentration recommended is something around 2% to 10%. So it varies in different protocols. But usually I think around 4 to 5% works very well. But I think for incubations, I think we dilute the serum again to probably 1% or 2%.
Okay. Sounds good. There are so many questions, which is wonderful. I'm sure if we included Sreethu's email in the follow-up email that we send to you guys, he would be happy to answer any other questions that you guys have. As I mentioned, we will be sending a follow-up email to you guys, and it will include the recording of this webinar along with the slides, a link for a free sample so that you can request a free sample of any of our primary unconjugated antibodies. And also you will receive a certificate of attendance for attending today. So with that, I just want to thank everyone for attending the webinar today, and a big thanks to Sreethu for sharing all his knowledge with us. And I look forward to seeing you all at future events. Thanks so much. Have a good day.
Thank you, everyone. Bye-bye. Thank you, Afrida.
Thanks.